Okay, so following last lecture on transcription, our next part is translation. So now actually taking the transcript that we created from yesterday, let me just highlight that. So we've made this transcript. Okay, so now it's mature mRNA. And now we're going to go through the process. Well, how does this become actual code? How does this become a protein? And that process is called translation, when you actually translate what your transcript is telling you. All right, so DNA translation, following the process of creating an mRNA transcript of code, the next stage is reading this code and creating the protein. So here we have the code, okay, and the translation part is now taking this code and turning it into the amino acid sequence that we have down here. Okay? And a couple things to keep in mind. Now we're going to go through this a little bit more in depth, but one thing I just kind of want to show you while we have this picture up is notice that the letters go in triplets. Okay? So they basically go in little groups of 3. Oops, I didn't mean to do that. Let me highlight. So letters are read basically 3 at a time. Right? So every time you see 3 letters, it codes for a specific protein. So AUG, those 3 letters in that order, codes for methionine. CCG, in that order, codes for proline, and so on and so forth. That's the general idea. Let's go through this a little bit in depth. So the keys to translation. The mRNA code sequence is broken up into 3-letter sequences called codons. Okay, so here's our mRNA sequence. Okay, I'm just going to get a nice yellow highlighter. So here's our mRNA sequence, okay, and the letters are just below. But notice they're broken up into 3-letter, what we call codons. Basically little blocks of 3 letters. That's how DNA gets read. It gets read at 3 letters at a time because 3 letters represents an amino acid. So GAG represents the amino acid glutamine, and CCC represents proline, and UCA represents serine, and AUC represents ulene. Okay? And basically the order of these letters, okay, gets corresponding to the order of the amino acids, which makes the different proteins. So the order of the codons determines the order of the amino acids and therefore the type of protein made. All right, becoming a little bit clearer when we do an example. Now, this little table over here, right, is a very important table. It's basically, it tells you what the amino acid is based on the letter sequence. Okay, so let's take, and I'm going to show you kind of how you use this table. So let's take a look at the mRNA sequence. So here's my mRNA sequence right here. Okay, so it's AUG, AAG, UUU, UAG. Now what we're going to do is we are going to decode this. So I'm going to do it at 3 letters at a time. So the first thing I look at is the first 3 letters. Let me do a better job of boxing those in. Oops. So I'm going to look at AUG, and I have to figure out those 3 letters, what amino acid does it code for? Well, we already know it codes for methionine, but how do we use this table? Okay. Well, the first thing we do is we say, okay, the first letter is A. Well, on this first column right here, okay, is where all your first letters are. So you go and you say, okay, well, my first letter is A. So now I'm only focusing on these columns. Okay. So all the other columns aren't important to me. Okay. Now, my second letter is U. So I have to go, okay, well, the second letter, if I go across here, some green, if I go across here, the first column is U. So now this is the only box that I'm concerned with. Okay? A was my first letter, U is my second letter. Now when I want to go to my third letter, it says my third letter was G. So I go and I go all the way across and I say, okay, AUG is methionine. There's my first one. Okay. All right. Now let's do the next one. So I'm going to erase this and say, okay, my next 3 letters are going to be A, A, G. So I'm going to go over here and say, okay, let's see where AAG is. Let me erase all this first. Say, okay, well, my first letter is A, so I know I'm going to be in this row. Okay, and my second letter is A, so I know now I'm going to go down and I'm now in this box. Okay. Then it said my third letter is G. So I go to the very bottom, and it's lysine. So there's two things that code for lysine. Whether it was AAA or AAG, it would both be lysine. But it's AAG across here, we go down, and there's lysine. Okay. My next one would be PHE. But I'll go through it coming up here. So this is the practice. This is what you kind of have to be able to do. Okay. So when you look at this, what we're going to go through is kind of basically the entire process. So if this is my DNA strand and it shows me half, then I know my other half is going to be the correspondent. Let me pick a better color here. T, A, C, C, A, T, C, G, A, T, T, G, G, A, and A. That's my DNA strand. Okay. Now, we have to decide, okay, well, if the gene is on the bottom half, so we're going to say the gene is on the bottom half here. So this is the code that I want to make the gene from. This is my gene right here. Let me go across, I'm going to highlight it and say this is my gene. So if I'm going to make my mRNA code, it's going to be complementary to this. Okay? So as I go through and make my mRNA, if that's a T, then I know that this, is going to have to be an A. And since that's an A, this is going to have to be a U, because we use U's instead of T's on RNA. That's a C, so this is going to be a G. That's a C, this is going to be a G. That's an A, this is going to be a U. That's a T, that's an A. G, C, U, A, C, C, U, U. So there's my mRNA. Okay. Now, don't worry about tRNA. Okay, if we had more time, if we were actually in class, we would go through tRNA, but just kind of block out tRNA right now. Okay, so now what we have to do is we have to read this code, this mRNA, and figure out the amino acid sequence based on that code. So what we're going to use is, so the first one is AUG. Okay, then we've got GUA, then GCU, AAC, and CUU. So we have 1, 2, 3, 4, 5 amino acids, and right down here we've got 1, 2, 3, 4, 5 amino acids. That makes sense. So the first one we got to figure out, well, where's AUG? And I put MET as my first amino acid. My second amino acid is GUA. Make sure my third letter was A. Back, and I write valine in here. So this is my growing protein. That's amino acid 1, that's amino acid 2. And we keep going. Now I have GCU, ALA. Then I've got so the amino acid ASN. And the last one is CUU, LEU. And this is what my growing protein is. Okay, so this is how DNA gets turned into mRNA that gets turned into an amino acid sequence, which is protein. Okay, so I'm going to go back to my table and I'm going to say, well, my first letter is A. So I go right here, and I know I'm in this column. So my second letter is U. So then I go in here. So now I know I'm in this column right now. And then my third letter is AUG, and it's telling me that it's MET. So I go back over here. Let me just erase what I had done before. And I said it was GUA. So my first letter is G. So I'm in this row. My second letter is U. And so GUA is right here. Codes for valine. I go, my first letter is G. My second letter is C. My third letter is U. GCU is right here, alanine. C. So first letter is A, second letter is A, third letter is C, ASN. So first letter C, second letter U, CUU is leucine. Okay. Now where all this happens, okay, this happens basically at the ribosome. Okay, now I told you don't have to worry about these tRNAs, so just kind of forget those codes here. Okay, but notice here's our mRNA. Okay? And the mRNA basically attaches to the ribosome. And that ribosome basically slides down the mRNA. And as it slides down the mRNA, it is basically taking all these amino acids that it's gotten banked. So there's a whole bunch of different amino acids all just kind of floating around in here. And based on that code, it will say, okay, well, the next three letters on the mRNA are UUC. Okay? Which amino acid needs to come in there to complement it? It needs to be AAG. When that goes in there, this phenolphthalein will then attach to this, and then I will get my growing polypeptide chain, or the protein. Okay? And then it will keep reading. The next three letters will read, the next three letters, and then we'll just keep adding and adding and adding until I get my protein. Okay, there's one more I kind of want to show you because this is a kind of an overall picture of the entire process, transcription and translation. So we have transcription up here. Okay, so inside here we're going to open up the DNA. The DNA is going to make some pre-mRNA, and then we're going to process it by cutting out those introns, okay, putting a KL and a TAP. Now we have the mRNA. The mRNA leaves the nucleus, attaches to a ribosome. So that's what this little kind of picture thing is. It's a ribosome. The ribosome then reads the mRNA, and based on that mRNA code, creates this polypeptide chain, which is a protein. Okay. Thanks, guys. So I know this was a little bit probably challenging to go through, but when I post some videos tomorrow, hopefully that will help some clarify.